everybody. Are we going live? Are we live yet? Are we live? What's going on? All right, looks like we are live. Cool. How's it going, everybody? Week 3, and we got this week and next week. Some people, your cells got contaminated and or died. We will be sending out new cells as soon as possible. Hey Kent, how's it going, man? Hope you're enjoying driving home from work. But yeah, we'll be sending out people new cells. If what's up, Dave? How's it going, man? How your cells surviving? Yeah, we'll be sending out new cells soon. It's just a process of growing them up and sending them out and everything again. We'll probably list them on the website pretty soon. So if you just want to purchase them, it'll be the easiest. We'll also list media and other things on the website. Glad they're growing well, Dave. Keep taking care of them. So that if you need more of these things, you can just purchase them at any time. Y'all are like, our first class. This is the first time any human beings have ever done this stuff, right? So it's all a huge experiment. So if you have stuff that goes wrong or you run out of stuff or anything like that, feel free to contact us. We're here to help. We're here to figure out how to make this work because, it should work. Jewel, don't worry about condensation. It's probably just caused by if you're using the heat mat. One thing you could do is you could stick the heat mat inside a cooler, a styrofoam cooler or something like that, and it'll keep the temperature more homogeneous across the whole culture. Yeah, Kent, we'll make calfectin and stuff available. We'll make everything in the kit available, so don't worry. But also, if you don't see it on the website eventually, just contact us. Y'all are a pretty hardcore group of biohackers, so we're here to help you out. Just contact us, and somebody will figure out how to get it to you. But yeah, styrofoam cooler, Jewel. So hopefully everybody's 12 well plates started to grow up a little bit well. I understand we really haven't done a tutorial on the microscope software yet. Some people, seem to be interested. So yeah, Jewel. Tinfoil might help a little, but it's a really good conductor. But it's also doesn't hold heat very well. All right, let's see if we can switch this around. All right, microscope. There's my microscope. Let's start up some Amscope. All right, Amscope MD300. All right, so we got the Amscope up and running. Let's get something up on there. All right. So one interesting thing is that my batteries haven't run out on this microscope yet, and I've been using it for a while. They last pretty long, the batteries in the scope. So first thing, what you probably might have noticed is that your plate does with this piece of cardboard because it was annoying to constantly have to hold the plate up to look at the corners and stuff like that. It's up to you what you want to do, but it makes your life a lot easier if you replace this stand with something else. Instead of cardboard, I would maybe try to use something that was flatter and something that was level, because this isn't level, so it can get kind of weird. But you know, it works. So that's what I do. So there's a couple things you'll start to notice when you use the microscope. One is that they have a couple things. So if you're not familiar with exposure and this little light compensation, if you're not familiar with exposure and gain in cameras, we'll help you out a little here. So exposure is basically what happens is if your camera, I don't know if it's the sensor or what, but if it's not getting enough light, what you can do is you can leave the sensor exposed to the thing for a longer period of time to collect more photons of light. So when you're changing the exposure, you can change it and it will change. Basically, it'll help brighten up the image or make it more clear if it's dark. It'll capture more photons. Now this thing has auto exposure, which usually works great. But you know, sometimes there's instances where you're going to want to do the exposure yourself. What I use more is the analog gain. So the thing with exposure also though is it's like an average over a period of time, right? So if you have stuff that's moving and you do a long exposure, then you'll see these little like trails, these blurs. That well is really overgrown, so don't judge me. So that's something that exposure. Now gain, on the other hand, just tries to multiply the light that comes in. Algorithmically or hardwarely or signally. So you can see when I turn up the gain, the brightness of the picture kind of starts to turn up. Now this is really good when you start to get to like the 20x, right? So the 20x aperture lets less light through. So what you have to do generally is bump up that gain a little bit to get a brighter picture of those cells. Ooh, look at those cells. Yeah, so bump up the analog gain, get a nice brighter picture. Now, auto white balance. Auto white balance is something that normally you won't modify, but what it's basically doing is it's kind of like taking out a lot of the color that is in these samples, right? So if you look at your well, it's actually pink in color, right? So when you're seeing something that isn't pink, what's happening is the software in the camera is trying to auto color the image, so to speak. Because normally this should be purple, pinkish type color. Let's see if I could find it. Where is it? Where's this purple, pinkish? So the interesting thing that I found with this microscope and camera is that the camera actually has a UV filter on the end of it. So you can actually look at GFP fluorescence with this camera. You can if it's bright enough, like in the GFP yeast we have on our website, you can actually see. Now, removing the auto white balance and removing the color, making sure it's not auto color temperature, you can actually see this a lot better. I've messed around with a little bit, but I haven't messed around with it a lot. I imagine if you figure it out just right, you can probably also see GFP fluorescence in these mammalian cells, though the expression's probably not as high. Now, a lot of this stuff, hue, saturation, brightness, contrast, a lot of this stuff is more just to make your pictures pretty. You're probably not going to use it all that much. Brightness, contrast, maybe a little. So generally, if you want to like bring out some contours and stuff like that of the cells, you know, up the contrast, and it'll help you see more of the contours of the cells and stuff like that. Color, grayscale, you know, like the difference is just obviously whether you're seeing it in color or grayscale. All right, so video and images. So if you click the snap button, it's going to take a picture of what is on your camera, and it should appear up here. So here's your camera, here's the image. Now, after you take an image, there's a lot of things you can do. You can actually process and do different things to the image, right? So you can look at changing different things, making it look better. A surface plot, that looks fun. The surface plot tells you where the look at where the cells are and stuff. Ooh. Yeah, there's a lot of stuff you can do. One thing, let's see. It's always good to do auto level, auto contrast. These things, you know, kind of make sure that across the whole image, the light level and everything is very similar. What is cool and interesting else on here? I don't know, but that's the image, right? And then from here, you can save the image that you want. Now you can also take video. The video on this stuff is not quite intuitive. Because it just kind of like runs. You have to set the time limit before it's going, which is kind of annoying, but it's pretty cool because what you can do is you can do time-lapse stuff and stuff like that, right? So you can take pictures over and videos over a long period of time. So if you're doing an experiment where you want to watch your cells doing shit, moving, growing, whatever, you can do it with this camera. But yeah, I don't like the fact that it has this, what happens if you take off the time limit? Will it just go until you stop it? Maybe. I mean, these cells aren't really doing much, so the video is kind of pointless. But you know, sometimes you got cells moving around and stuff like that. A lot of different stuff. It's good for a lot of different reasons. Record your video, upload it somewhere, save it for posterity. Something good to use. All right. So we got, no, I don't want to say, how do we just close this? Close. Do you want to save changes? No. All right. And back to our image. All right. So we got our image. Confluency, right? So we were talking in some of this stuff about confluency. Now, generally, people do a lot of stuff. So this is a close-up. What we probably want to do to look at confluency is zoom out a little bit. Let's zoom out to the 4x. Cell counter. What about cell counter? Yeah, you can get, if you're trying to count cells over a specific distance using like a micrometer or something like that. So what we did send, but I don't know if we'll have time to cover in this class, is it's tryptophan blue staining, live dead cell staining, which helps you count living or dead cells. The thing is, most stuff you won't really have to worry about that we're doing. So it's you should be pretty okay, but we sent you tryptophan blue so you can do live dead cell staining if you want. I don't know if we included the protocol in the classroom yet, but there's plenty of protocols online. It's pretty easy. All right, so here's our camera, and here's our thing. Now, you can judge this however you want, right? But for me, this well is overgrown, right? It looks like it's probably like 80 to 90% grown. And that's too much. Generally, you probably want to be about 60 to 70%. So you should keep up with your cells all the time when you're doing an experiment in order to work with them under the best possible conditions. Now, for this experiment, what we're going to do is we're just going to kill the cells or try to kill them using antibiotics. So it's not the end of the world if the cells are overgrown or something like that. What you want to do, though, probably, is before you start this experiment, go around and take a picture of each well that you're going to experiment on, so you can see before and after pictures of your experiments, right? It's just easier to keep track of, especially if you have little experience with this stuff. Otherwise, you might be like, I wonder what that, I can't remember what that's, well looked like when I first started this experiment, so it's hard for me to compare, right? Well, something like this well looks terrible. It's got too many cells. Another thing to remember is, for instance, if you look at my plate, if you get a well that's contaminated, just remove all the media in it and just let it die. Like, don't keep messing around with it. You can still use the rest of the plate just fine if one well is contaminated. That's not a big deal. So don't worry about that well. Remove the media. Just let it all die and dry out and whatever. These are wells, too overgrown. Sorry. But the first thing that you want to do before you do any experiment generally is you want to change the media, if you haven't recently. So generally, you probably want to change the media every 3 to 5 days. If you're doing like a genetic engineering experiment, you probably want to keep that closer to 3, just so that the cells stay healthy. So when you're doing antibiotic selection, every 2 or 3 days, you want to change out the media and add new antibiotics. It's just good to have fresh antibiotics in there to keep the selection pressure on. So make sure that you change out your media. If you're just letting something grow or doing like what we're doing, you're going to test out antibiotics on these things, then it doesn't matter too much. But, you want to try to replicate your experiments as closely as possible. So do what you got time to do, right? So what we're going to do, let me see. This class is, let's go to classroom. Antibiotic screening. All right. Antibiotic screening. So when you get new cells or when you're performing an experiment on cells, there's there's three main antibiotics used in mammalian cell culture. The first is G418, which is pretty popular, and then peromycin, and then like hygromycin or something like that. Now G418 is used with like every organism. It's a pretty good antibiotic. Hygromycin is used with plants and stuff. Peromycin, I've never really seen it used besides with human cells, but whatever. Anyway, if you've never tested your cells before, you want to test them with the antibiotics. This is because depending on the environment, the temperature, the media, how well your cells are growing, etc., antibiotics are going to affect them differently. You want to find out what antibiotics work for you and your cells, and not just in general. Because a lot of times what happens is that in general, what somebody writes on the internet or somebody used for another protocol is probably, in my experience, is not the same as what I've had to do. Understand that like most other cell culture, especially in academic papers, is done with DMEM, which is a different media than L15, and it's grown with carbon dioxide to help buffer the media, which is just ridiculous. I don't get it. And so these cells are going to be subject to completely different and usually they're in pretty high-end incubators, so the temperature doesn't fluctuate much. It's like 0.1, 0.2 degrees, around 37. So they're going to react, respond, experience the world completely differently, right? That's okay. It just means that you have to figure out how your cells are responding to the environment. And to do that, we're going to do a G418 screen. G418 is the antibiotic that kills these cells, and what we're going to use for our genetic engineering screen. The plasmid that we're using, the FST plasmid, it contains a neomycin G418 resistance cassette, so that the goal is to just get the cells to survive. That's it. Get the cells to survive. It's a basic transformation, and it teaches you how to engineer these cells using plasmids, right? First, we have to screen the cells to figure out what concentration of G418 we're going to use for our genetic engineering experiments. Now, generally, the concentrations I have found that work well have been between 400 and 600 micrograms, but it might be different for you. Don't just follow what I see. Follow what you actually see. It's really important, and it's not that complicated, right? So let's go to. So the first thing you're going to do with your plate is you're going to replace all the media in it, right? And so take a sterile filter, take a sterile syringe and a filter, take your L15 media and filter it, and this time put about 1 ml in every well. The 1 ml just makes it easier to calculate. 800 microliters is fine if you want to do 800 microliters like we did before. But yeah, 1 ml is perfect because when we add the G418, it allows us to add it 1 microliter per 200 micrograms. So we're going to set up a plate, and we're going to try to make it something like this, right? So we're going to screen a bunch of different concentrations of G418. Now, each microliter of G418, so you're going to be using the 0.5 to 10 microliter pipette. 1 microliter is about 200 micrograms, So what you're going to do is for every 200 micrograms, you're going to use 1 microliter, right? We have 1 ml of culture. It's 1 microliter per ml. That's what our stock solution is. So we're going to do 1 microliter per ml of G418 for every 200 micrograms. So 400 micrograms would be 2 microliters, 600 would be 3, 800 would be 4, 1,000 would be 5, 1,200 would be 6, 1,400 7, 1,600 8, 1,800 9, right? And that should give us a full range of concentrations of G418 across the board. Now, the CTRL is control. We also want to do control experiments here, right? So the control experiments are not adding any antibiotic. So what we're going to do is we're going to see how the cells with antibiotic compare to the cells without antibiotic. What we should start to see is that the cells with antibiotics start to die off. Now, what you need to do is you need to make a spreadsheet, something like this, right? Concentration and percent cells that have died. And what you want to do is you want to track this for, about a week. And over the course of this week, what you want to find out is what is the lowest concentration at which the most cells have died, right? And this is really subjective. Generally, you'll see most of the cells dead, 99% in a lot of cases. And as you extend this over more longer days, that the number of wells with 99% will increase. You'll have more of them. Generally, you're going to do your genetic engineering experiment over the course of about a week and a half. So you're trying to see which cells have like 99% death at about 5 to 7 days. After that, it's going to be pretty much the same. So grow out your cells, and on day 3 or day 4, what you're going to do is you're going to replace the media with fresh media and fresh antibiotic. So you're going to just take out the old media, add in new media, and then re-add the antibiotics again, right? Generally, this is what you're going to do with selection every 2 to 3 to 4 days, depending on how fast your cells are growing and what's going on. You're going to replace the media and add antibiotics. Now, generally, when you replace the media, you want the media to be, room temperature or warmer. If you can get it up near 37 degrees, that's great. But you want the cells to be as close to temperature, the media to be closest temperature to the cell's normal habitat as possible, right? You generally, you can just turn honestly, make sure you have a waste container to collect the cells and bleach them or whatever, because, I don't even know if the human cells can survive any place, but that's common laboratory practice. But yeah, what I just do is I just turn the whole thing over. Just make sure you do it one fluid motion so that the media from a well doesn't splash into another well. But generally, you can just flip it over and dump out the old media. If you want to be anal, use a pipette and remove it one by one. It's up to you. And re-add the media. Generally, after you add the media, people let the cells sit for like an hour or two. They're pretty stressed out. You change the media, the temperature might be very different. The cells might be experiencing a little bit of, I don't know, shock, a little stress. That's it. Probably experiencing a little bit of stress, so you want them to, like, when you actually test them or work with them, not to be so stressed out. So it doesn't hurt to add the media and let them sit at 37°C for an hour or two. And then add the antibiotics, or then do the next experiment on them. But I can't say I've seen too much of a difference. So it's up to you, up to how anal you want to be, and what you're going for. Sometimes there's no correct or incorrect. Both work equally well. So once you let the cells rest, or once you add the media, then just add the G418 directly to the well. So you're going to pipette, 9 times. So just make sure you switch pipette tips between pipetting, and make sure you add the correct amount of G418 per well. It's a good idea to label your plate. No matter if you have a diagram or whatever online, just label your plate, just so you know. Track it. So every day, take it out, look at all the wells under the microscope, check and see how many have died. And this is totally a guesstimation. Now, the way I figure it out is the cells that are living adhere to the plate, right? So if we're looking at, let's see, something like this. Yeah. Let's see, something like this. Generally what you want to do is you want to not shake, you want to like swirl your plate a little bit, and then look at it under the microscope. When you swirl the plate, what's going to happen is you're going to see how many cells are attached and how many cells are detached, right? So the detached cells will move around on the screen. Let's see if we can see this. So you can see the detached cells moving around, and the attached cells just staying in the same place. It's a good way to get an idea of how many of the cells are dead. If you have very few cells that are still attached, then you know that most of them are dying. If you have lots of cells still attached, just mark it down. So what you're going to want to do is just estimate this stuff. You don't have to be exact. It's the only important thing is that you're consistent with your estimates, right? So if you're like, if there's like very few cells attached, I'm going to say like 90. If I can only see like one cell attached in the whole screen, I'm going to say like 99. If it looks like half the cells are attached, just be consistent, right? The one thing you're majorly just looking for is like, have the majority of cells died? So are there very few attached cells anymore, right? And you should see a lot of the cells moving. Now the thing is when you replace the media, understand that a lot of those detached cells are going to get washed away, right? So a lot of the detached cells are going to get washed away. If you want to make it more clear which cells are detached and which cells are attached, you can use PBS or something to rinse out the cell wells. So add some PBS, swirl it around a little bit, and then dump it out. or add some PBS and pipette it up and down a little bit and then dump it out. After that, you should have most of the dead cells gone. Each time you do this, it should remove more dead cells. It can help a lot with determining the living and dead cells, but like over time, after you see it a few times, it shouldn't be too difficult just to tell by yourself. So keep your spreadsheet or something written down. Make sure you have an idea of how many cells are dying and at what time period, because this will be key to the genetic engineering experiment that we do next. Right? So this week, what you're going to do is you're going to take your plate, you're going to replace the media, you're going to add antibiotic, and then you're going to watch it. After, 3 days, you're going to replace the media and the antibiotic again, and you're going to continue watching it and recording it in a spreadsheet, right? And then next week, when we do our genetic engineering experiment, you will have the opportunity to use that information to help you genetically modify your cells. Will the suspended cells be different shapes than the attached cells? Hey Leslie, sometimes, but sometimes it's really hard to tell. If you read the first two guides, they talk a little bit about different cell shapes and what suspended cells versus attached cells look like, but sometimes it's not so obvious. Because cells can get stressed. It depends on how many cells there are and how overgrown they are and all this stuff. It's generally just a good guideline that if the cells are detached, they're dead. So stick with that. If they die, if they're looking ugly and they're still attached, they'll eventually die probably. So and when they die, they detach. So just go on the basis that if the cells are detached, they're dead. All right. Any questions? Any questions about what you're going to do this week? So just be sterile, clean everything up, right? Always wear them gloves when you're handling your cells, and clean those gloves. Take care of those cells. We are going to ship out more cells this week, so hopefully everybody who killed their cells should get some fresh cells. Or, maybe it wasn't your fault. Totally okay. I get it. I've killed lots of cells, so I understand completely. But we should be sending out more. And we'll start to list the stuff for this class on the website individually, so then you could just buy it. Jewel, what if you run low on media? We like I said, we'll start to list some of this stuff on the website so people can start buying it. If you need more media, just email us right now, and we'll do our best to get it out to you. But hopefully you don't use it all up. Yeah, 2 tubes is should be plenty to finish the experiment. So each tube contains 15 ml. So for the last 2 experiments, we're going to be working in these 12 well plates, right? So, well, actually, you might need a little bit more. Yeah, just contact us. We'll send you some more to keep those genetically engineered cells growing. We'll send you another 4 tubes or so, or whatever. Just contact us. What we'll do is I'll put something up on the website, where people can just order it. I'll give you a code. I'll post it on the Google Classroom. So I'll post a code in the Google Classroom so you all can order some of this stuff. Sound good? Any other questions? Any other questions before we finish up today? All right, if there are no more questions, then I'm going to take off. Remember, if you got questions, post in the Google Classroom, contact me. I try to keep up with that stuff as much as I can. If it's really important and you need it immediately, just let me know. Victoria, what if it turned yellow and cloudy? That means they're contaminated. We'll send everybody. We'll post a link in the classroom where people can just order new cells, so we don't have to do this manually. Dave, what's the doubling time of HEK 293? It really depends on the conditions. I don't think people have really grown it a lot in the media in the way we're growing it. But generally, you'll probably hit confluency in like 3 days, 3 to 5 days, 70% confluency. So that's, it could be faster. Depends on how many cells you seed with. It depends on a lot of stuff. So, yeah, if your cells turn yellow and cloudy, it's contaminated. So I'll post a link tomorrow in the classroom and you can order more. Anybody else? Anybody else got any questions? Any other questions? All right. Well, let's talk again next week. Even if the class ends, we'll still be around to answer your questions and help you out with everything. So don't worry. Don't freak out. After the class is over, everything gets shut down. The Google Classroom is going to stay available and everything like that. So, if you're having trouble or your cells are dying or getting contaminated, everything should be okay. Otherwise, I will see you in the same time next week for our last class. We'll teach you how to do the genetic engineering part. And you should have all the skills in your arsenal then to hopefully be able to do it yourself. All right, talk to you soon.